Okay guys, so last time we talked about natural selection, evolution. That was chapter 19 and chapter 21. Now we're going to go to chapter 22, which is going to be how did life actually arise? And we're going to go over some different mechanisms that not only tells us how, the first organisms came about, but also how different species came to be. So we talked about Darwin. We talked about the Galapagos Islands. He found that on these Galapagos Islands there was plants and animals found nowhere else on Earth. One of the things they found was one of these birds called a cormorant, and it's flightless, but it's on the Galapagos Islands. So if it's a flightless bird on an island, that kind of makes things, puzzle you a little bit. Because if it's a flightless bird, it did not come from the mainland. It didn't, fly over. If it's a bird of flight, we can emigrate very easily. But if it's a flightless bird, what that tells us is that it is native to that island. So how did this bird come to live on the Galapagos Islands? Well, what happened was, before this bird was there was very low or very restricted gene flow with other flightless birds on that island. And because of this, we had speciation. Another way we could say this is we had a macroevolution event. When we talk about macroevolution, guys, and we'll talk about this a little bit more, macroevolution is like giving rise to a whole new species or a whole new subsection in an evolutionary tree. That's a macroevolution event. Microevolution is just like editing a specific species. But if you guys remember from last chapter, if we have restricted gene flow, just like what happened in this bird here, what's going on is the genetic variation is getting smaller. So what that can do is that can actually lead to speciation, meaning that two new species come about. So that's what happened at the Galapagos Islands with this flightless cormorant bird. So like I said, it leads to speciation. We have one species splitting into two or more. Now, when speciation occurs, they have a common ancestor, and that common ancestor is going to go extinct for the new species to come about. And again, guys, it's not going to be one species morphing into another species. It's going to be one species morphing into two or more species. So we already talked about microevolution and macroevolution. Macroevolution, we are getting new species out of it. We are getting a whole new group in an evolutionary tree. Microevolution, we got a population, we got a species, and we are just editing them. We're changing the alleles, and then we are letting natural selection act on those. All right, so section 1, guys, things that biologists study. Morphology, this is going to be comparing living things and all of their structures that they have. So comparing one organism structures to another organism structures to see if they might share a common ancestor. Physiology, that's going to be the study of their body, how it moves. Biochemistry is all the chemical processes that are going on in an organism. And you'll find that a lot of the organisms, especially like in the animal kingdom, the biochemistry is going to be very, very similar in them. So we're going to go over some species concepts. The first one is the biological species concept. So what this does is it distinguishes two species based on the degree of genetic exchange between their gene pools. So when we have the biological species concept, and we're talking about that, let's go back to the liger. I know we talked about the liger. We could even talk about a mule from last chapter. They are sterile. Are they really considered a species based on this? No, they're not, because they cannot have genetic exchange between two different organisms, two different animals' gene pool. All right, so if we have two mules mating, nothing happens. They're sterile. So there is no genetic exchange. All right, so The whole thing with the biological species concept is it's based on the potential to interbreed rather than like physical similarities here. So you guys can see all these people look very different, but they can all breed together. We don't need two people with red hair to breed to produce fertile offspring. All these people, their genes are close enough together where they can breed and make fertile offspring. So the next thing is on reproductive isolation, and this is a very broad term. We're going to go into a lot of different parts and examples of reproductive isolation, but overall, just a general broad examination of reproductive isolation. It's the existence of some kind of barrier that impedes two species from making fertile offspring. Hybrids are the offspring of crosses between different species, so we can produce hybrid organisms, but most of the time, these hybrid organisms are going to produce offspring that might not be very fertile, or they might be sterile overall. So we're going to go over prezygotic barriers and postzygotic barriers. So these are two different parts of reproductive isolation. So two different examples. First one, prezygotic barriers. What this means is we are not going to have fertilization. Sperm, egg might come together, but the egg is not going to get fertilized for whatever reason. All right, so we're not going to have fertile offspring, obviously, if we do not have fertilization. Okay. So there's three different types of prezygotic barriers. There's habitat isolation, temporal isolation, and behavioral isolation. So here's habitat. They do not encounter each other, okay, not that much. They live in different habitats, so they might not have like a physical barrier in between them, but maybe, like a lot of organisms, they're going to stay in their habitat because it's safe. They're not going to go over and mate. So it's not really like a physical barrier, but they're still not going to get to the other person. So think of this one, guys. Let's go over flies. Some flies just may live on the water. If you guys have ever been fishing or anything, you'll see flies all on top of the water. Some might live on the water, and some might live on the soil next to the water. Well, if they like to live on the water, they're probably not going to go and mate with the bugs in the soil. The flies in the soil, same thing, they're not going to go to mate with the flies on the water. All right, so that's habitat isolation. Second one, temporal isolation. Species breed at different times of the day, different seasons, or different years, so they are not going to breed. An example of this, an American toad, so just two different toads here, they are going to mate in early summer, and then there's another toad called the Fowler's toad. They mate in late summer. So one mates in early summer, one mates in late summer. It's kind of like, the one's not in the mood during certain, times of the season. So they are not going to be mating just because of their different times. All right. Yeah, I went over the frog one, so we'll just skip this one. All right, last one is behavioral isolation. So there's courtship rituals. If you guys have ever seen there's like some birds that do some very, very odd dances. I'll try to find a video and post one for you. But there's a blue-footed booby, if you guys have ever seen that bird before. And it does a very interesting mating dance. But what will happen here, guys, is some of the mating dances will, work and, it attracted the female. Some might be like, the female might be like, what is this? This is not what I'm looking for at all. So if that is the case, there's not going to be, once again, any kind of mating or fertilization occurring there. So like I said, guys, I'll post the video. I'll try to find one of the blue-footed booby, and I'll post that for you. Okay? All right. So two others, guys, for prezygotic barriers. There's mechanical isolation and gametic isolation. So the first one, mechanical, there are some type of difference. So in their morphology, there's some type of difference that prevent successful mating. For this one, maybe they're just not matching up. Okay, we got the two Snells in there. But maybe, their shells are a certain way and they're not matching up. So there's not going to be any fertilization there. Second one is gametic isolation. Sperm of one species may not be able to fertilize eggs of another species. So if you're keeping organisms apart when they are fertile, that's an example of gametic isolation. This is how they allow dogs to keep their purebred bloodline. You don't want a Rottweiler and an English bulldog together when the bulldog is fertile. They will actually separate them if it's two different species. If they're trying to keep the purebred bloodline, they'll separate them when they are fertile to make sure they're not reproducing. All right. Okay, so moving on to postzygotic barriers. So what this is going to do, postzygotic, is something is going to happen after fertilization in order for a fertile child to not be produced. Okay, so when we have our postzygotic barriers, there's three we're going to go over here real quick. So the first one is reduced hybrid viability. So we have these two different species. They might not be far apart, or they could be. If they are far apart, and I'm talking about with amino acid differences and things like that, guys, if they are far apart, it's probably going to be a prezygotic factor where they are not going to have fertilization. They might not even mate, all right? If they are able to mate and they are able to go through fertilization, they might have a postzygotic barrier. So, first one, genes of different parent species may interact and impair the hybrid's development or survival. So, whenever and if we do humans, okay, we have 23 chromosomes from the sperm, 23 chromosomes from the egg. When they come together, all those chromosomes are going to mix, are going to interact and form that first diploid cell. So when they, in this example here, when they interact together, they might actually be bad for the new organism trying to be produced, and that organism might not survive. Second one is reduced hybrid fertility. So even if the hybrids are good, so they're born, they look fine, they look healthy, they're able to do basic, fulfill their basic needs of eating, sleeping, that kind of thing. They still might be fertile. So that means they're not going to be able to have offspring of their own. An example of this would be, if we go with the donkey example here, we got a donkey, we got a horse. When they mate together, they form the mule, which, yeah, it might be a nice workhorse, but it is sterile, so it's not able to pass its genes further. Last one is hybrid breakdown. So let's say we produce a hybrid offspring. They're fertile, so they're able to also produce offspring. But the offspring they produce are very, very weak, they're feeble, and they might themselves actually be sterile. Okay, so we are done with that part, guys. All right, we're done with the prezygotic and postzygotic barriers. So this will be our last slide on the biological species concept. So I'm just going to go over the last three here. With the biological species concept, we pretend that gene flow is not happening. So we want speciation to happen. If there's no gene flow, there's a good chance speciation could occur. So an example of this is going to be, we have grizzly bears, we have polar bears. When they mate together, they can actually form a hybrid called the roller bear. Kind of strange looking thing, right? All right. So moving on, we're done with the biological species concept. We're going to go to the next one, which is called the morphological species concept. So why we use this, the morphological species concept, it's going to be used if we find all these new species and they haven't been discovered yet. So it's just like a blank slate. We've got all these new species, we haven't discovered them yet. It's like, okay, well, let's start putting them into groups here. So what this is going to do, the morphological species concept, is it's going to allow us to separate them into different species. Some might look really similar to others. We could test their DNA, but if we're just going simply by morphology, looking at them, we can put them into different groups by, okay, look, this one looks just like this one. We're going to put them together in a species. So that's pretty much it. Next one is the ecological species concept. This is going to be viewing a species in the terms of its ecological niche. So what's going to go on here, guys, is we are going to see what its purpose is in the ecosystem to which it lives. In every ecosystem, guys, when it started off, there was holes in it. We always got to have producers and consumers, and we got to have a bunch of different consumers so that we have birds eating the bugs and something eating the birds and so on and so forth. If you just have, let's say, a bear eating bugs, that's not going to be enough food to support a bear. We need an organism in between so that the bear has enough food to live. So the phylogenetic species concept, this is pretty much taking a species or more than one species and grouping it into the smallest group we can find, putting it on a phylogenetic tree, and there's your species. This is kind of not as investigative as the other ones, in my opinion. So I'm not going to touch on that one too much. Okay, so we're going to go over two different types of speciation, how it occurs. Again, remember, guys, we don't want a lot of gene flow in order for speciation to occur. So we're going to go over allopatric and sympatric. I'm going to do allopatric today, and then we will call it a day. So here's some fish. All right. We have the fish up top that are all orange, and they're able to swim from one side to the other side, and everything is good. But then the water goes down. The fish that are left on this side all become one species, or stay the orange species. The other ones, something happens where they go through speciation, and now they're this green color of fish. So after the water level increases here, we now have the fish intermingling, but now they are two different species. So this is an example of allopatric speciation. We had a geographical barrier between them, and that caused speciation to occur. So while we're on this, guys, let me talk about below, what happens below this area here. Now, we had our species of fish here. If I wanted to look in the rock strata below, we would find a lot of fossils buried with the different fish. Now, if we wanted to go the same distance down on both sides, so it'd be the same layer of strata, we would find those species, whatever species of fish that are probably really closely related to one another. But if we just keep going lower and lower and lower, we would find that strata that I just talked about is going to be much different than the ones below it and below it and below it. And if we went to the absolute lowest point, we would find the most diversity, the most genetic variation, the most genetic divergence between that and that rock strata I just talked about there. Okay? That was just kind of a review for you guys from a previous chapter. But, okay. So allopatric speciation. This is due to geographic isolation between a population. So an example of this, in the Galapagos Islands, we probably had one of our flightless birds, this one here, it probably originally was a bird of flight. It came by the Galapagos Islands. It probably enjoyed itself. It was a nice ecosystem for the bird of flight to live in. But then it didn't use its wings. It didn't need its wings because it was cool where it was at. So it actually probably speciated, and since it wasn't using its wings, it lost the ability to fly. And then it stayed there. Maybe something happened where there was geographic isolation for this bird to speciate. Probably it could have. It could have been a number of other things, but that is our best guess. All right. So with the geographic barrier, our main thing is we got to be able to know if that population is able to disperse, if it is able to intermingle with the other population. If it's not able to get to the other population, then they probably will become two different species over time. So as those two different species start to go through speciation, okay, so we have some geographic barrier, we have the species, it now speciates, we got two different species. Each one of those species, so the one on the one side, the one on the other, they are going to then go through microevolution. They're going to go through mutations. They're going to go through natural selection. They're going to go through genetic drift, where they are going to drift further apart from one another and actually become their own species. And later on, if they were to try to mate, if they are so far apart from one another, that mating might not actually be possible. It might be a prezygotic barrier after that. What caused that? Well, the geographic barrier between them caused that. All right, so here are two different fish guys. You can see they look about the same, but something happened where there was a barrier in between them. Now, we got this one on this side, and has a different body shape where it's not able to swim for a long time, but it's able to go for rapid bursts, swimming very, very fast for a short amount of times. And the other one over here is able to swim for a long time, but it's not able to go real fast. Why? Well, what happened was some of them got preyed on, some of them did not. If they get preyed on, they want to be able to swim very fast. Might not be for a long time, but just long enough for them to get away. This one over here, under low predation, it's just going to casually take its time swimming here and there, so on and so forth. So, all right, I think this is our last slide on, almost, allopatric speciation. So, they were doing another study, guys. They found a bunch of shrimp, and they separated them based on where they lived at. So they found that back a while ago, it was probably all one area, and then the different islands started to form, and so the fish or shrimp became isolated. Okay, and here's kind of what happened here. We had this area here, and we have our different shrimp on both sides. All right. So this one is kind of common sense, guys. Regions with many geographic barriers. There's a lot of barriers between species. They usually have more species based on these geographic barriers. So maybe those geographic barriers weren't there the whole time. Maybe the Grand Canyon, that's a good example, it wasn't this big canyon we see today. So when it was less of a canyon, we probably had less species. But then we had this big barrier come about, and we had a lot of speciation occur there. All right. So when we talk about intrinsic, when you guys see that word, barriers to reproduction are intrinsic. Separation itself is not a biological barrier. So what we mean by intrinsic, we can use for this example, is they have different reproductive times or different reproductive ways or something like that. But overall, there is some kind of reproductive barrier stopping them from reproducing. Okay, so next time we're going to go over sympatric speciation. We're done with the allo speciation. We're done with that one. So that was our geographic isolation one. And we'll come back next time and go over the other one. Have a good rest of your day, guys.